I now call to order the Society's 2379th meeting in the for the 86th Joseph Henry Lecture in the 146th year since the Society's founding in 1871. Good evening, everyone. My name is Larry Milstein. I'm the president of PSW, the oldest scientific society of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members and guests to tonight's lecture on exoplanets and the search for life. We'll begin with a few announcements and a reading of the minutes of the 2378th meeting and Tony James's lecture on malaria, CRISPR, and gene drive. We'll then turn to this evening's lectures, followed by a question and answer period. Thereafter, I will present a small thank you gift to our speakers, make a few closing announcements, and then adjourn the meeting to the social hour. Let me start out by noting that as part of our annual celebration of Joseph Henry, the Society's first and longest serving president, I will be using the Society's ceremonial gavel. It's made from the timbers of the White House, recovered from the Truman reconstruction that took place in 1949 to 1952. The timbers were originally used in reconstructing the White House in the period 1815 to 1817 after the British, with whom we now have a famously special relationship, burned it to the ground in the War of 1812. Our speakers tonight, which will be introduced later, and the subject of their lectures. And please join me in thanking the sponsors of the Fall 2016 and Spring 2017 lecture series, the Policy Studies Organization, in cooperation with the American Public University, and a generous anonymous donor. In addition, tonight's Joseph Henry Lecture is sponsored by Millen White, Solano, and Brannigan. You may wonder why a law firm is sponsoring a science lecture. Well, I happen to be a partner at that law firm. My slides are not quite in order. I am very sad to report that longtime General Committee member, former president and dear friend, Bill Maish, passed away. Bill was a physical chemist for all of his professional life and worked at the Naval Research Laboratory for many years. He was a member of PSW for more than 50 years, and he served the Society in many roles over that time. He was president of the Society in 1979 and was a member of the General Committee in one capacity or another from the time I joined PSW, about 12 years ago, until this year. It's a small consolation that we were able to honor and celebrate Bill earlier this year at the 2374th meeting on 24 February, and we'll greatly miss him. I am pleased to announce that the following new members have been elected. Karen Jones, Clinical Director at Lantern Mental Health Center and an attorney interested in astrophysics, cosmology, and genetics, who found PSW on the web. Dennis Miller, CEO and President of Science and Technology International, interested in molecular biology, astronomy, physics, energy, and environmental sciences and policy, who comes to PSW through friends in the Cosmos Club. And tonight's speakers, Debra Fischer, Professor of Astronomy and Geology and Geophysics at Yale University, Hannah Ruth Wakeford, postdoctoral fellow, Science and Exploration Directorate at NASA Goddard Space Flight Center, Shawn Domagal, scientist at the Sciences and Exploration Director at NASA Goddard SFC, Aki Roberge, astrophysicist in the Exoplanets and Stellar Astrophysics Lab at NASA Goddard Space Flight Center, and Mark Clampin, but he's already a member of PSW. Please join me in welcoming them to the Society. All new members and members who did not previously get a copy are invited to come up to the front of the room and get their personally autographed by me copy of Volume 1 of the Bulletin of the Philosophical Society of Washington, where you can read why the Philosophical Society of Washington is named the Philosophical Society of Washington, and understand a little bit why we are trying to kind of convince people to call it PSW or PSW science. It has always been about science, but back then, philosophy meant natural philosophy to these founders, and natural philosophy was the term for science. Recording Secretary Preston Thomas will now read the minutes of the previous meeting's lecture on malaria, CRISPR, and gene drive by Tony James.
At the Cosmos Club in Washington, D.C. on April 28, 2017, President Larry Milstein called the 2,378th meeting of the Society to order at 8:11 p.m. He announced the order of business and welcomed new members. President Milstein presented a summary of the 37th meeting of the Society held in 1872. The minutes of the previous meeting were read and approved. President Milstein then introduced the speaker for the evening, Anthony A. James, a distinguished professor in the departments of microbiology and molecular genetics and molecular biology and biochemistry at the University of California, Irvine. His lecture was titled, "The Mosquito, Synthetic Biology, CRISPR, and Malaria." Dr. James began by explaining that malaria has afflicted humans for all of recorded history, with references to deadly recurrent fevers appearing in Mesopotamian cuneiform in 6000 BC. The famous Greek physician Hippocrates noted the link between swampy waters and fever cycles. Malaria is a blood-borne infectious disease resulting from a protozoan parasite that is transmitted by mosquitoes. Today, there are 5 species of malaria parasite that affect humans, the newest of which crossed over to humans within the last 15 years. The 2 species most deadly to humans are Plasmodium falciparum and Plasmodium vivax. Importantly, there are no free-living forms of the parasite. It exists only inside a suitable host. Malaria is only spread by adult female mosquitoes, which feed on blood as a source of protein for nutrition and egg production. Malaria today is endemic in the poorest countries in the world, existing in all tropical and subtropical regions. In 2015, there were 212 million cases worldwide, resulting in approximately 429,000 deaths, 90% of which were in sub-Saharan Africa. Although we have many tools that are effective against malaria, including bed nets, spraying, eliminating standing water, and antimalarial drugs, for various practical, economic, and political reasons, these tools have been insufficient to control malaria in many countries. To date, no effective vaccines have been developed against any protozoan parasite, including malaria. Dr. James then described a promising new strategy of engineering mosquitoes with genes that prevent the transmission of the malaria parasite. To do this, scientists first search for mosquito genes that are activated when the mosquito feeds. They found suitable genes related to digestion. Next, they identified a gene in mice that reacted very strongly to human strains of the malaria parasite, neutralizing it. Splicing this mouse gene into a mosquito creates a transgenic mosquito that expresses antibodies in the midgut and the salivary gland every time the mosquito feeds, curing the mosquito and eliminating transmission back to humans. After developing a malaria-immune mosquito, the next task is to distribute these genes throughout the mosquito population. This can be done simply by breeding and releasing millions of mosquitoes, but a more effective strategy is to include a gene drive in the engineered mosquito's DNA that ensures that the antimalarial genes are preferentially inherited when the mosquito breeds, overriding the traditional Mendelian inheritance patterns. The creation of a gene drive is possible because of advances in the use of CRISPR-Cas9 gene editing system, which consists of the Cas9 nuclease, which cuts DNA, plus guide RNA, which targets the cuts to a precise position in the mosquito DNA sequence. When the cell attempts to repair the cuts to its DNA, the most available model for reconstituting the cut section is the lab-created plasmid containing the antimalarial mouse gene. Moreover, this engineered sequence also contains the coding for the Cas9 nuclease and guide RNA, which means that the repaired DNA will copy itself autonomously to the other unmodified chromosome, creating mosquitoes that have a 100% chance of passing along the engineered genes. Dr. James explained that in addition to the functional parts of the engineered plasmid, the nuclease, guide RNA, and the malarial resistance sequence, it also contains a protein for red coloring derived from a jellyfish that allows engineered mosquitoes to be readily distinguished from wild mosquitoes. Dr. James noted that drugs, insecticides, and vaccines all have well-established regulatory pipelines from discovery to development to delivery, but genetics-based vector control does not. Nonetheless, Dr. James expressed confidence that carefully constructed, rigorously conducted phased trials will demonstrate to regulators, governments, and communities that genetically engineered mosquitoes are a safe and effective malaria control strategy that, when combined with existing tools, could ultimately enable World Health authorities to finally eradicate malaria. After the conclusion of the lecture, President Milstein invited questions from the audience. One questioner asked how the mouse gene confers malaria resistance in the mosquito. Dr. James explained that the engineering confers two forms of resistance. First, it creates antibodies that bind to surface proteins on the parasite uses to invade host cells. In the second method, it simply coats the surface of the parasite in antibodies, preventing it from interacting with the host cells. Another questioner asked why the engineered mosquito uses genes from mice antibodies and not human antibodies. Dr. James explained that human resistance acts only during the human blood cell stages and does not provide complete protection, so the mouse gene was chosen to target malaria completely and at the point of transmission. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. At 9:59 p.m., President Milstein adjourned the 2,378th meeting of the Society to the social hour. Temperature, 24°C. Weather, partly cloudy. Attendance, 105. Respectfully submitted, Preston Thomas, Recording Secretary.
Thank you, Preston. Are there any questions, comments, corrections, additions to the minutes? If not, I will entertain a motion from a member to accept the minutes as read. Bob Hershey has the motion. A member second. Eric second. All members in favor? Aye. All members opposed? Minutes are accepted as read and will be posted to the website in due course. We now turn to tonight's meeting and the 86th Annual Joseph Henry Lecture, which will be in 4 parts. It's my pleasure to introduce the topic of the lectures, Exoplanets and the Search for Life. Just a few decades ago, the idea of planets orbiting stars far from our sun was a commonplace only in science fiction. In scientific circles, their likelihood was debated, with views tightly bound to theories about how stars and solar systems form. In the 3 decades since the first reliable report that an exoplanet had been detected, we have seen an explosion of exoplanet identification, fueled conceptually by knowing that they actually exist, and practically by vast improvements in methodology, computational capabilities, and instrumentation. As of April of this year, 3,607 exoplanets have been detected in 2,701 distinct planetary systems, including 610 multi-planet systems, approximately. We know that current techniques cannot detect most of the exoplanets that might exist around all the stars we can observe. And we are also just beginning to detect properties of the exoplanets that we can observe beyond their mass, orbital periods, and distances from their suns. We've recently had our first observations of exoplanet atmospheres and a few indications of their composition. Just today, in fact, we read of a publication on the watery atmosphere of an extraordinary Neptune-like exoplanet authored by one of our speakers tonight. Can I name it? and her colleagues. Eventually, soon actually, we hope, we will have sufficient resolving power and analytical tools to survey a statistically significant number of exoplanet atmospheres to have our first empirical understanding of how many Earth-like planets there are that have atmospheres similar to our own, and how many of these contain gases that we associate with life. This series of 4 lectures will survey our current knowledge of exoplanets, explain the capabilities and limitations of current detection techniques, discuss the TRAPPIST-1 system of planets, explore prospects for improving our abilities to study exoplanets, and ways we may be able to find indications of life, and at least tentative answers to the question, are we alone? Our 4 speakers on these topics will be Debra Fischer, Hannah Ruth Wakeford, who is the author of the aforementioned paper that came out, I think, this week, Shawn Domagal Goldman, and Aki Roberge. And here to introduce them before their talks is Mark Clampin, Deputy Director, Sciences and Exploration Directorate, NASA Goddard Space Flight Center. Please hold questions for the question and answer period at the end of the lecture. We'll have all of the speakers on the stage, together with Mark, for free-ranging answers to your queries.
Okay, good evening. So tonight's talk, as you heard, will be on exoplanets and the search for life. And we have 4 speakers who are all very outstanding speakers and are heavily involved in this field and have made many of the sort of breakthroughs in this field. So let me start. Our first speaker tonight is Professor Debra Fischer. And Debra is a professor of astronomy and a professor of geology and geophysics at Yale University, and an adjunct professor of physics and astronomy at San Francisco State University. Debra began hunting for exoplanets in 1997, and she has discovered hundreds of extrasolar planets, including the first known multi-planet system. Among her many honors and awards, Debra was elected to the SFSU Alumni Hall of Fame, was a Cecilia Payne-Gaposchkin Lecturer, and has been elected to the American Academy of Arts and Sciences and to the Connecticut Academy of Science and Engineering. Debra earned a BS and an MS in physics from San Francisco State University and a PhD in astrophysics from UC Santa Cruz. Our second speaker tonight is Dr. Hannah Wakeford. Dr. Wakeford is a NASA postdoc fellow at the Goddard Space Flight Center, shortly to be a 2017 Giacconi Fellow at the Space Telescope Science Institute. Hannah's work focuses on characterizing the atmospheres of exoplanets through the observations with the Hubble Space Telescope, which is now in its 27th year of operation. And Hannah is the recipient of the Robert H. Goddard Award for the individual scientific achievement and her work on the TRAPPIST-1 planets. Hannah is an advocate for science communications, and she has won a number of awards and commendations. During her PhD, she produced and hosted a weekly live radio show and was a regular expert guest on BBC Radio Devon and a number of British TV news programs. She's also appeared on a number of TV shows for the BBC, including BBC Horizon. Hannah earned a BS and an MS in planetary and space physics at Aberystwyth University and a PhD in physics at the University of Exeter. Our third speaker tonight will be Shawn Domagal Goldman. Shawn is a research space scientist in the Planetary Environments Laboratory at NASA's Goddard Space Flight Center. He focuses on comparative planetology, and as a member of multiple interdisciplinary teams, he simulates the atmospheres of other worlds, including those of ancient Earth, modern Mars, ancient Mars, and other exoplanets. Shawn is a member of the Curiosity Science Team, the XOS Science and Technology Definition Team, and the HABEX Science and Technology Definition Team. He's also a member of the NASA Astrobiology Institute's Virtual Planetary Laboratory and the Rocky 3D Resolving Orbital and Climate Keys of Earth and Extraterrestrial Environments with Dynamics. Our final speaker this evening will be Dr. Aki Roberge. Dr. Aki Roberge is a research astrophysicist at NASA's Goddard Space Flight Center, and her research focuses on observational studies of planet formation around nearby young stars. In addition, she's deeply involved in developing future space missions and currently serves as a study scientist for the large UV optical infrared surveyor mission known as LUVOIR. This is a highly capable multi-wavelength future space observatory with ambitious science goals. Dr. Roberge received a PhD in astrophysics from the Johns Hopkins University, and after completing a bachelor's degree in physics with a planetary science minor at MIT, she became involved with space mission development during her postdoctoral work at the Carnegie Institution for Science and later at the Goddard Space Flight Center. So without further ado, let me introduce the first speaker, Professor Debra Fischer.
One of the things I hope to do tonight is give you a sense not just of how we find planets and what the limitations are, but why I do this, what keeps me up at night, what inspires me to keep working on this for such a long time. I started working in the field, as you heard, in 1997. At the time, there were 3 exoplanets that we knew about. Thanks. And have been incredibly fortunate and lucky to be able to work in a field that's just exploded underneath of me. So I'm going to tell you a little bit. I want to start by giving you a sense of our place in space and time. This might be something that you guys are familiar with, but I think it's something that I always like to reflect on. And if after all these years I still find it amazing, I hope that you will as well. We think of here we are on this beautiful planet, right? But if we look, our planet is just a pretty small, insignificant little speck of a planet compared to Jupiter, Saturn, the other planets in our solar system. And of course, here's the real view of our planet. You see it, right? Everybody sees it? Okay, it's this little dot here, suspended in a beam of sunlight. This is a picture of the Earth that was taken by the Voyager spacecraft as it was going into the outer part of the solar system. And as Carl Sagan wrote, look again at that dot. That's here, that's home, that's us. On it, everyone you know, everyone you ever heard of, every human being who ever was lived out their lives on a mote of dust suspended in a sunbeam. This is the kind of picture that if we work very hard, we might be able to get of planets around other stars. Our sun is just a very ordinary star, right? It's fairly unremarkable. It's one of billions of others in our Milky Way galaxy. If you think about the most massive stars in the galaxy, there are 1,000 stars like the sun compared to those really rare, really massive stars. But the most common type of stars in the galaxy are not like the sun. They're much lower in mass. They're the tiny M-dwarf stars. I put up a picture of the moon, and on it you can see a little frame. So we're going to focus in on this trio of CCD detectors from the Hubble Space Telescope. But I put up the moon because everybody knows how big the moon is, right? I want to really instill exactly what it is that you're seeing and give you this sense of scale. So now I'll take the moon away because when the Hubble Space Telescope was looking at this part of the sky, the moon wasn't there. This is a zoom in now on those detectors. And Hubble looked at this part of the sky because astronomers thought it was interesting that there was nothing there, right? It's just, you can see right here a few stars, but otherwise a pretty empty field. And the question that astronomers had is, what is there in space when we think that there's nothing there? And I just can't imagine what it would have been like to be at the Space Telescope when the answer came down. And we saw this image of 10,000 galaxies in this tiny point of the sky. Now, there's nothing special about this part of the sky, and Hubble has proved that by looking at other points of the sky. The entire celestial sphere is wallpapered with these other galaxies. Each one of them has hundreds of billions of stars in them, right? So that is a fairly staggering view of our place in space. If you think about our place in time, it's not much better. The universe began with a Big Bang 13.7 billion years ago. That's a number that I don't have any sense of, that, right? Human beings don't have any experience with 13.7 billion years. But Carl Sagan wisely knew that we all know what a year feels like, and so he rescaled 13.7 billion years to 1 year, the cosmic year, and on January 1st, the Big Bang occurs. By May, the Milky Way galaxy forms. By August, on this cosmic calendar, our sun forms, and along with it, all of the planets in our solar system. By September, our planet is crawling with life. Microbial life dominates the world still today, but lives in September, October, November, and December. And we really have to zoom in to see where we are. Here we are in December now, and it's Christmas Eve when the dinosaurs appear. And the dinosaurs are here for a whole cosmic week on this cosmic calendar, right? It's incredible. So what did they do in their cosmic week? I often wonder, like, where are their telescopes? What things, great robots, and what things did they invent, right? Did they have CRISPR technology, for example? So it's a little humbling that we don't really appear until the last seconds. You have to zoom in. And in the last 60 seconds of the year, we start to see human beings, humans as we would recognize them. The earliest Homo sapiens appeared on our planet a few minutes ago. All of human history has been in the last 60 seconds. So when I talk to the Yale students, I tell them, sure, you can study history, study 60 seconds of the cosmic year. Sure, you can study geology, study that little speck of, that little mote of dust suspended in a sunbeam, or you can study astronomy, right, and get to study all of this stuff. Even more sobering, technological humans really have only been here for a millisecond. But what we have done in this millisecond is incredibly profound, right? We've figured out that there have been other species on this planet before us and that they went extinct. We figured out why they went extinct. We've built telescopes. We've learned that there was a Big Bang and that the atoms in our body came from the nucleosynthesis in the cores of other stars. We really are a remarkable species, and this we've done in a millisecond on the cosmic calendar. What if we're here for a year, for even an hour on the cosmic calendar, or like the dinosaurs for several days? I have absolutely no doubt that we will be out into the galaxy exploring space, because that seems to be part of who makes up part of who we are. So we don't dominate the universe in any sense, and I want you to be thinking about this as you listen to my colleagues tell you about the missions that we're going to be doing. Our place in space is small. Earth was home to even the dinosaurs for a much longer time than it's been home to us. But again, we're a remarkable species. We stand on the threshold of learning which of the planetary systems around nearby stars harbors life. Biochemists are teaching us about the origin of life on Earth. And I would say if my view on this has changed a lot over the decades that I've been teaching. I used to wonder if life could have been just this random chance event that occurred on this planet. And now I think that life was actually an inevitable consequence of the processes that occurred on this planet. And if that's correct, that has profound implications for the probability of life elsewhere. So I remember being at a conference in 1992, and astronomers had been looking for planets for some time and not finding them. And I had the Star Trek view that there must be planets, there had to be. And so why couldn't we find them? And a friend of mine, Dimitar Sasolov, who's a professor now at Harvard University and a theorist, said patiently, Debra, so stars, of course, form in clusters, and the more massive stars will go supernova and photoionize protoplanetary disks. And it could be that most stars, in fact, don't form with planets. So that was our view in 1992. It was sobering. We had to admit that that was a possibility. And then in 1995, the first exoplanet was discovered around a nearby star. Oops, that went by quickly. Kind of like, let's see, maybe it's because Clinton was running for president and that went by quickly too. There's a different Clinton. Because people wondered, is anybody out there, right? That was it. It wasn't that we had found a rock orbiting another star. It was this re-energized this question of whether or not there could be life elsewhere. And that's what keeps me up. That's what keeps me going. because I have no doubt that there is life out there. I have no doubt that there is life on hundreds of nearby planets orbiting hundreds of nearby stars. And our job is going to be to find it in the next 20 years. So the techniques that we use, this is the technique that I use with my team, is called the Doppler technique. We never even see the planet, right? But we believe in physics, and what we see is the host star right here. We take a spectrum of the star, spreading the light out into its component wavelengths of light. You see these little absorption bands that come from the atoms in the atmosphere of the star absorbing light, so that light is missing. It's that missing light that as the star comes towards us and goes away from us, that missing light, those wavelengths shift back and forth and allow us to track the velocity of the star. And once we know the speed of the star, the velocity of the star over time, we can deduce the presence of a planet that we don't even see, orbiting stars that are hundreds of light years away. It's a pretty spectacular feat. Let me give you some sense, because I sort of feel like this slide makes it look like it's easy. These spectral lines that you see span tens of pixels on a CCD, the charge coupled devices that take pictures now in your camera. And as these lines go back and forth, our current precision allows us to measure a shift in the line with, anybody want to guess, a half a pixel, a tenth of a pixel? No, our precision now is a thousandth of a pixel. We can measure shifts in these lines to 1/1000th of a pixel. What we're trying to do in the next few years is actually take that an order of magnitude further to 10 centimeters per second. And that's because that's the speed that the Earth induces in the sun as it orbits the sun. The other technique that you're going to hear a lot about tonight is the transit technique. So here we look at distant stars. We don't resolve them like you see in this picture, but we measure the brightness of the star, and we see that the brightness dims. Right here, we're measuring the brightness. Brightness dims because that planet is blocking out some of the light of the star. Now, the depth of that transit dip is proportional to the radius of the planet divided by the radius of the star, quantity squared, because it's just a ratio of the area of the two cross-sectional areas. So you can see we can figure out how big the planet is, because if you know the radius, then you know the volume of the planet, 4/3 pi r cubed. And with the technique that I use, the Doppler technique, we measure the mass of the planet. So if you put the mass together with the volume of the planet, you can calculate a bulk density. And you can figure out if that orbiting planet is a rock or if it's a big puffy gas giant planet. Again, planets we never even see, we never set eye on, and yet we're able to deduce all of this incredible information. In the last years, we've gone from 1992, no planets, to today, many planets. And these plots sort of show a history, track the history of what's happened. On the left-hand side, you see the planet mass, which is shown in units of Earth masses. So here's one Earth mass. There aren't any planets that are down there, so I felt very safe putting the legend over that part of the plot. But we found the pink dots all show the radial velocity technique, and we found lots of planets that are like Jupiter and some Neptune-like planets. And then in blue are the transits that were detected from the ground. In 2009, NASA launched the Kepler mission, and the situation changed dramatically. Now you see all of these yellow dots. So isn't it odd that here again is the mass of the Earth, right? What Kepler is really doing is telling you the radius of the Earth, but we can, make an educated guess about what the mass is. And then here's the orbital period of the Earth, 1 year. And you can see that there aren't a lot of planets in this part of space. It's as if nature is conspiring against us to hide these guys. In fact, what happened is that Kepler lost 2 of its reaction wheels, right, and had to be repurposed into another mission. I think if Kepler had continued, these detections would have rolled out in time. But Kepler transformed exoplanets. It told us that most stars have planetary systems and that small rocky planets like the Earth were common, ubiquitous, I would say. So we went from thinking that most stars don't have planets, to today, where we realize that in fact almost every star forms with planetary system. There are billions in our galaxy, and with that thought, life may be more common than we thought. We haven't found real Earth analogs yet. These would be rocky planets that have water on their surface, but we're still looking. Why search for more planets? Well, that's one reason. We want to find more planets that are like the Earth. The technique that I use, the Doppler technique, hasn't found habitable worlds. Kepler is studying planets that are 1,000 light years away, too far to follow up. So my team is now building an instrument called Express. It's a spectrograph that is the culmination of my career. Everything I learned in the last, thank you over dinner, everything I learned over the last 20-some years has really gone into this instrument, and we're commissioning it this year, so I'm very excited about it. And then from Express, we'll bridge forward to future missions. I'm not going to read you the fine print here. I'm really happy to talk to anybody about this instrument, though, and all the unique features that we're putting into it. We'll be delivering it to the Discovery Channel telescope, which is in Arizona at Lowell Observatory, and we'll be carrying out the 100 Earths project to search for 100 Earths orbiting nearby stars. So Express is going to measure the masses of transiting planets, double the scientific return of those missions, and then other large space telescopes that you'll hear about next will attain spectra of the atmospheres that we discover and look for signs of life, biosignatures. I'll end with this slide just saying that I think this has an enormous impact for society. This is a philosophical society, right? It answers the age-old questions about whether there are other worlds like our own, what our solar system is like, and whether there could be life elsewhere. Thank you.
Well, that was a fantastic introduction into the different types of ways that we look at planetary systems. And what I'm going to do specifically is talk to you about the transiting planetary systems and how we can delve deeper and understand more about those worlds by looking for signatures in their atmospheres. So, as was shown before, we have a transiting planet, and what you can see in that top figure is that same image, but the difference between an Earth-sized planet and a Jupiter-sized planet. A Jupiter-sized planet will block out a lot more light than an Earth-sized planet, and we're measuring this relative change in the amount of light from that star, equivalent to seeing a mosquito fly in front of a street lamp, where you're seeing 1% of that star's light being blocked out. Now, what we're also able to do from this is look at the atmospheres. And we can do that by getting a spectrum of this transit. So if we look at the transit, the amount of light being blocked by that planet, over lots of different wavelengths, we can build up a spectrum of the starlight that has been filtered through that planet's atmosphere before it reaches our telescopes. So we're looking at starlight, which is passing through an alien planet's atmosphere before reaching our telescopes, and being able to measure the different molecules in the atmosphere by how they block it differently. So each molecule, lucky for us, has a unique fingerprint, and we can measure those unique fingerprints in the atmospheres of these strange worlds. So I'm going to talk to you a little bit about the history of what we've been doing with the Hubble Space Telescope, looking at these giant hydrogen, helium, Jupiter-sized gas planets, and take you through the technique and the measurements that we're making. And then I'll move on to these Earth-sized worlds in the TRAPPIST-1 system and what we're doing for that. So the Hubble Space Telescope has been an amazing workhorse for the last 27 years. It was launched before exoplanets had even turned into science fact. So it wasn't designed to do these kinds of measurements. But we've been using it for the last decade or so to look at exoplanets and trying to understand their atmospheres. And what I'm showing you here is a study, and this is the actual data that you can see up here, and I'm not sure where the laser is on this, at the top. So this is actual data from the Hubble Space Telescope, where we're measuring the spectrum of these different exoplanets. This is 10 Jupiter-sized exoplanets that orbit very, very close to their star. So they're incredibly hot. They have very large, puffy, extended atmospheres. And we're seeing that starlight filtered through those atmospheres before reaching the Hubble Space Telescope. And if we look at the change in the amount of light over all of the wavelengths, we can see the evidence of different molecules in that planet's atmosphere. And what you can see here, from the top to the bottom, is water absorption features right here. And then as you go down, they start to be encroached by clouds in the atmosphere. Now, these clouds are nothing like you would think of here on Earth. They are exotic, they are high temperature, and they actually form rocks in our own planet. These are magnesium silicate clouds, these are aluminium oxide clouds. These are clouds that are made of the strangest rock and gem material here on Earth as liquids in these planets' atmospheres. And I want to show you some simulations we've done of the sunsets that you would see on these planets. And if you look, HD 209458b was the first planet to ever have its atmosphere studied in this way. That's our beautiful green sunset at the top. And the reason for this is the sodium in this planet's atmosphere. And the sodium would cause that planet's sunset to look very green and alien. Now, down in the bottom corner is HD 189733b, and you can see that the sunset looks very similar to what we have here on Earth. But the scale of these planets is completely off. The scale is because they're so close to their star, the star itself takes up a huge amount of that sky. And the reason why we're seeing this beautiful earth-colored sunset in this atmosphere is because it has particles in its atmosphere which scatter the light in the blue and not in the red. So it's very similar, but very different materials than the Earth. In the Earth, it's the nitrogen in our atmosphere, and in this planet's atmosphere, it is liquid droplets of glass. WASP 12b is my favorite of these. I hated it when I was doing my PhD because the data is very difficult, but this planet's atmosphere is amazing. It gives you this weird film noir grayscale sunset. But that's because in its atmosphere, it has large particles of corundum. Corundum is rubies and sapphires. Clouds in the atmosphere causing this dull gray sunset are actually the base components of rubies and sapphires that we see here on Earth. These places are amazing, and they will blow your mind every time you think about them. And we're learning so much more about how planets can form and what their atmospheres and nature might be like. And this is just a snippet of all of the work that we've been doing with the Hubble Space Telescope. Massive smattering of data for you there. And this represents roughly 40 exoplanets out of the over 3,000 that have been discovered, that we've been able to study their atmospheres. And that is increasing every single year. We have a huge program on the Hubble Space Telescope to look at 20 more exoplanets. So over the next 2 years, we're going to be getting so much data. But that brings me to, these are giant planets. What about smaller worlds? Now, the TRAPPIST-1 system was in the news a lot earlier this year, so I'm going to take you through this really exciting system and hopefully inspire you into learning more about exoplanets. To look at smaller worlds, we need to look at smaller stars. And as Debra said, the smaller stars are more abundant in our universe. That's very helpful for us. But they're much harder to look at because they're small and they're faint. They're in the red. But what's also really great about these smaller stars is that as your star gets smaller, it gets colder, and it pushes this temperate zone where liquid water can exist much closer to its star. And the closer the planet is to its star, the easier it is for us to be able to detect multiple transit events. And the more transit events we can see, the more times we can try and look at its atmosphere. Now, the beginning for the TRAPPIST-1 system was May last year. Just a year ago, this planetary system was announced where the Belgian telescope, TRAPPIST, discovered TRAPPIST-1, a ultra-cool M-dwarf with 2 and possibly a 3rd planet orbiting it. And what you can see here are those detection transits. So TRAPPIST-1b, the largest and innermost planet, TRAPPIST-1c, and then this strange light curve here. This is multiple planets passing in front of the star all at the same time. And we took the opportunity on May the 4th last year to point Hubble at an event, a special event that only occurs every 2 years. We looked at these 2 planets, B and C, as they passed in front of that star at the same time to search for signatures of a large hydrogen envelope around these planets. Now, the reason we're searching for this is because small planets could maintain lots of hydrogen around them in an envelope. Hydrogen acts as a greenhouse gas and will change the environment on that planet. So we're trying to understand whether or not these planets have that envelope around them. From this observation with the Hubble Space Telescope, we found that B and C do not have this hydrogen envelope around them. And that's really interesting. That takes us to further studies trying to understand what their atmospheres are like. And it actually it tells us that these are likely terrestrial in nature, i.e. they are rocky, and they have compressed atmospheres. But then we got another exciting thing from the TRAPPIST-1 system. 500 hours of Spitzer Space Telescope time observing this planetary system. And from that, we discovered that the third planet in that system was not one planet, wasn't two planets, it was 5 planets. 5 planets in addition to those 2 inner ones that we'd already looked at. And you can see in this amazing video here, this is the real data from the Spitzer Space Telescope passing underneath. And this is a simulation of those planets as they pass in front of that star. So we're able to detect 7 planets in this system. Not giant planets, not like our solar system. These are all Earth-sized worlds. 7 Earth-sized worlds orbiting one star. That's a gold mine for the type of work that I'm doing. 7 planets around one star takes the star out of the equation. We're then comparing the planets to each other and comparing their atmospheres to each other. That tells us huge amounts about the impact of different planetary interactions and stellar interactions. But also, because this star is so small, it's an M-dwarf. This temperate water zone, liquid water zone, is much closer to its star. And if you place it in the context of our solar system, the TRAPPIST-1 system is 40 light years away. But if you place the planets in the context of our solar system, they all fit inside the orbit of Mercury. So they fit down in this tiny little gap right here. All of the planets, 7 Earths, that close to the star. And at this distance from the star, we see this transition from the hot inner part of the system to the middle temperate part where the water can turn into a liquid and pool on a surface if there is a surface. And then further out where you get the ices forming, out by H. So even though this is much closer to its star, it's actually because the star's cooler. They're in exactly the same zone as our terrestrial planets. Amazing, amazing system. And actually, we are currently looking at these 4 planets here, which span this temperate water zone. We're looking at those with the Hubble Space Telescope now to try and determine if they have this hydrogen envelope around it. The further away from the star, the more likely that that planet can maintain that hydrogen envelope. We can learn about that and that process with 7 planets, roughly the same size, roughly the same mass, all around the same star. It's a fascinating, fascinating puzzle that we are really working to try and figure out. And that takes me forward to the era of the James Webb Space Telescope. And just to give you a little bit of information about the James Webb Space Telescope, it has a 6.5 meter primary mirror. That is the largest telescope that will ever be launched into space. It's so big, we don't have a rocket that can take it there. It has to be folded up. So it is made of 18 segments, plated and covered in gold. Enough gold to make a nice-sized golf ball. It has 7 times the collecting area of the Hubble Space Telescope, and this will launch in October of 2018. And it has a nominal lifetime of 5 years. It operates in the near-infrared and the mid-infrared, whereas the Hubble Space Telescope operates in the near UV, at the optical mostly, and the near-infrared. And you can see in this figure at the bottom here, the size of the Hubble Space Telescope's mirror compared to the James Webb Space Telescope. This is an unbelievable feat of engineering, and it happened just near here at the Goddard Space Flight Center, which is unbelievable. It's also not going to be orbiting the Earth like the Hubble Space Telescope. It's going to be 1 million miles away at the L2 point. And this is a gravitational stability point, which allows it to be pointed away from the Earth, so we don't have to deal with the Earth's heat and the Earth's light. Now, what is Hubble going to be looking at? The James Webb Space Telescope going to be looking at? This is a simulation of planets that the TESS satellite, which will be launching in March next year. TESS stands for Transiting Exoplanet Survey Satellite. And TESS, we expect it to be able to find bright stars with planets that are small around them. And what we can see here is, oh, that was me. What we can see here is the radius of that planet. So this is using the transiting method. So the radius of that planet, up to 5 times the radius of the Earth, and the distance that planet will be, that star will be from us. And what you can see down here in orange is what we expect TESS to be able to discover for us to explore, and in blue, what Kepler has discovered. And what is absolutely brilliant about this is right down here in the best spot with the largest circles, and the circles mean that it's much easier for us to do this detection, is the TRAPPIST-1 system. They are the perfect candidate for us to point the James Webb Space Telescope at and try and understand their atmospheres. The James Webb Space Telescope is going to give us huge amounts of information. But what also wasn't expected is Hubble to last this long and be such an amazing workhorse for transiting exoplanets. The combined power of the Hubble Space Telescope and the James Webb Space Telescope will get us observations from the UV through the optical all the way out into the mid-infrared. This gives us information on the water content of an atmosphere, the carbon dioxide content of an atmosphere, the carbon monoxide content of an atmosphere. We can understand huge amounts of stuff about a planetary system. And this is just one simulation that we have of a giant exoplanet with one single observation for each of these colors that you can see here. This is the signal that you would be able to get for a giant planet. Now, what I'm showing you here is simulations by Natasha Batalha, who's also at Goddard Space Flight Center and will be defending her thesis very soon, of the TRAPPIST-1 planets. This is taking the example of TRAPPIST-1F, which sits happily in its temperate water zone. And this is a simulation of water-rich atmosphere, a nitrogen-rich atmosphere, and a carbon dioxide-rich atmosphere. And what these little bars at the top are showing you is after 1 year of observation, so 10 transits, this is the error bar that you will get with the James Webb Space Telescope. And as you go through and get 10 more with each successive year, how the error bar goes down. And if you look at the differences between each of these models, you can see that after the second cycle of the James Webb Space Telescope, we'll be able to understand whether or not these planets have a H2O-rich atmosphere. That's something that we never thought was possible. And it wasn't even thought possible when they were designing the James Webb. So this is going to be a fantastic era for exoplanets, our understanding of the environments of other worlds, which gives us the big question we want to answer. How easy is it to form a solar system like our own and a planet like our own? And what are the environments we should be expecting for other planets? Thank you.
Well, thank you for having me here tonight. I'm quite happy to be following that talk by Hannah, Dr. Wakeford, and the previous talk by Dr. Fischer, because I've been awed by the ability of my colleagues to understand and characterize the astrophysical nature of these worlds, as Dr. Fischer talked about, and to start to understand their chemical nature, as Dr. Wakeford just talked about. And part of what's so exciting about these TRAPPIST worlds is they may provide us with the first opportunity to characterize them from an astrobiological standpoint, to do chemical characterization on worlds that are small enough and aren't too hot or too cold to be able to harbor life. But that's not where I'm going to start. I'm going to start this story here with the Allen Hills meteorite, also called ALH 84001. This is a piece of rock that started its life on Mars, got ejected off the surface of Mars by an impactor, hurtled through space to land in Antarctica here on Earth, where it was discovered by scientists who took it back to their laboratory, put it under a microscope, and found that stuff. And they called the president. Well, they found some other things too. They found some chemical signs of life and some mineralogical signs of life, and they called the president, and they called a press conference, and they said, "We are not alone." And I was graduating high school the year that was announced. And in the 20 years since then, it was 1997, I think the astrobiology community would not say that this rock contains evidence of life. I think at best you could say there are things in this rock that are consistent with the past presence of life having been there, but there's nothing here that would rule out a lack of life on that rock. And I think to myself 20 years from now, having spent my entire life dedicated to the search for life on exoplanets and characterizing them for astrobiological properties, and I don't want to be in a situation where my colleagues or people from outside our field say that we did not find signs of life when I spent my whole life working on a mission that would look to find it. Unless you think I'm picking on the folks that worked on the Allen Hills meteorite, this is not the first time this has happened. You can go all the way back to Schiaparelli's craters back in the early 1900s. You can go back to Viking, the first time we did a direct search for life as an agency at NASA on another planet. And Gil Levin, the person who designed and led the instrument that made this measurement, which looked at the radiolytic release of carbon. They basically took radioactive carbon to the surface of Mars and gave that as nutrients to the soil and basically waited to see if that radioactive carbon and the nutrients baked off as gas, and sure enough it did. They got a positive result. And Gil Levin, the PI of this instrument, to this day will tell you that he found the first evidence of life beyond Earth. I will also say that I don't think the astrobiology community in large agrees with his assessment. Unless you think I'm picking on the Martians, this has also been a problem through Earth history. If you want to know what the earliest signs of life here on Earth are, you could go to the literature, and by the way, if you want to see outstanding and sometimes pointed scientific debate in the literature, look at the microfossil story for the earliest evidence of life on Earth. These are putative microfossils that another researcher called dubio fossils because they did not believe that they constituted the earliest evidence of life on Earth. So the point to this, before I get into the search for life on exoplanets, is almost everywhere we've looked, and in almost every way we've looked, it's been easy to find signs of life. What's been difficult is to rule out the ways in which non-biological processes can make the things that we're looking for that we think could only come from life. And so we go to the pale blue dot that Dr. Fischer showed earlier. And this is the data. These are the data that we're talking about getting. A spectroscopic signature, okay? Just photons from that one pale blue dot. We won't get spatial resolution, at least not with the first generation of telescopes we do this with. But we will get spectral resolution. We'll be able to spread that pixel of light out on a detector array based on its color, almost like it went through a super high technology prism, to see what colors are there and which ones are not. And based on the colors that are absent, we're going to say certain gases are in that planet's atmosphere. And if we find oxygen or ozone, and if we find methane, and if we find liquid water, we say, okay, there's life there, because life makes oxygen, and oxygen does not like to stick around in the presence of methane. And so you could only replenish that oxygen if there was life on the planet. No, you could only have that oxygen there if there was something like life replenishing it rapidly. Now, I still think that story stands, but when we talk as astrobiologists to instrument designers or engineers, they might say, well, just tell us what wavelength you want to find the oxygen, and we'll design an instrument to go and get that wavelength of light. And so just give me your two gases and the wavelength range that I need for them, and then I'm going to go design an instrument to see them. As an Earth scientist and someone that knows that story of the false positives, I think, well, it's not that simple. You have to think about the context. So I'll come back to this a bit later. And I think about the TRAPPIST systems when I think about context as well, because if we want to do a search for life, the lesson for me from the Allen Hills meteorite on Mars is that you had to interpret the data, those fossils and the mineralogical data and the isotopic data in the context of the rock. Well, for exoplanets, our context is going to be the entire atmosphere. It's going to be the entire planet, in a sense. This is a simulation of real data from the MAVEN spacecraft that's in orbit around Mars right now. And it's in a special orbit where it can look at things escaping from the Martian atmosphere, specifically ions of many things. This is a map of oxygen. And what happens is as Mars gets hit by a stellar wind, by the high energy radiation released from the sun, that can ionize the molecules in Mars's atmosphere and lead them to be stripped away along magnetic field lines. And it can also impact the magnetic field lines, which can accentuate that process. And I point this out because habitability is going to be something for exoplanets where the context of it is not some rock, it is the entire planet in the context of its star and also the neighbor planets. So I worry about this process in particular for the TRAPPIST worlds and other worlds we're going to look at soon for signs of life with the James Webb Space Telescope, because that process might be more efficient on planets around the cool type stars that have observational properties that will allow us to do this soon. So this is a map, or a diagram of, this is the mass of a star on the vertical axis, and the distance of a planet from that star on the horizontal axis. And the colors represent how efficient we think atmospheric loss might be for a planet with a given orbiting distance from a different star type. And as you move to the lower left, you get towards hotter and hotter colors. The white space is stuff that's just dead to begin with, right? So it's outside the habitable zone because that planet's either too close to the star or it's too far away. So the stuff that's colored is the Goldilocks zone. And as someone that came up thinking about the habitable zone for much of my career, it was often just thought of in those terms. Are you too hot? Are you too cold? Are you too big? Are you too small? The leading research in the field now is thinking beyond that, not just thinking about too hot, too cold, too big, too small, but like just what's the context? So here the context is also the star. Okay, what's the star doing? It's making that atmosphere be blown away. And that's more prevalent as you get to the lower left part of this diagram. So here's the TRAPPIST worlds that we're just talking about. The ones in the habitables are sitting right in that red area, and red means atmospheric loss should be quite effective. Now, I'm not saying we should not look at those worlds. In fact, the nerd in me wants to look at these worlds specifically to test this theory, because this is everything on this diagram now is a model, and this is an artist's interpretation of data, okay? And so if for no other reason, I want to test the theory that goes into that model by looking at these TRAPPIST worlds, even if they don't have atmospheres. To me, that's fascinating. And if they do have atmospheres and our models are wrong, then we've got a chance. That's a lottery ticket, right? We might be able to actually search for life in the next 5 years if that's the case. Now, there's another caveat, which is even if the atmosphere is retained, these cooler type stars, because there's the energy distribution of photons from the host star is different and weighted towards the higher energy regime, it also affects the chemistry inside the atmosphere. And it does it in a way that could lead to the buildup of oxygen and ozone in the atmosphere without any biological flux of oxygen into the atmosphere. And there's two reasons for that. The first is that high energy radiation that I showed stripping away the MAVEN atmosphere, if it doesn't strip away all of the atmosphere from one of these worlds, it's going to strip away the lightest atoms in the atmosphere. And the lightest atom is hydrogen. Now, hydrogen and oxygen are kind of like the moral combatants in chemistry, at least in terms of atmospheric chemistry. If you look at the history of our atmosphere's chemistry over the entire history of Earth, it's a story of the redox state, of essentially the balance between oxygen atoms and hydrogen atoms. If you get rid of all the hydrogen atoms, but not the entire atmosphere, you're basically left with oxygen atoms to dictate the chemistry. You'll end up with an oxygenated atmosphere. And I worry about that. So if the atmosphere is not lost and you just lose part of it, you're preferentially going to lose the things that would lead to an accumulation of the thing that you think is a biosignature. And the other reason is those photons are also really good at splitting up carbon dioxide molecules. If you split up CO2, you strip away an oxygen, and you let that oxygen hang out free, not seeing any hydrogens in the way to combine with another oxygen to form O2 and O3. So I worry about this from the, let's call it the Alan Hills perspective. So we've taken this into account over the last, really this stuff goes way back, decades. But what we've been doing lately is trying to come up with an observational strategy that accounts for not just the false positives I just talked about here, but all the false positives for life that we've thought of to date. And at the end of the day, it comes back to the same story. If you want something which has been called thermodynamic disequilibrium in the atmosphere, I actually think it's better described as kinetic disequilibrium. In some ways, thermodynamic disequilibrium is bad. I think of that as a free lunch. If you've got too much energy sitting out there, the analogy of one of my colleagues uses is if you've got a dish of candy outside your office at a university and it's not eaten, it's probably summer and the students are gone, right? Free lunch is something students don't pass up, and free lunch is not something that life passes up either. What you really want to look at is how is it that gas is retained in the atmosphere? Does it require life? And would you predict life would generate that gas at those rates? And this is our observational strategy. I won't go through the whole thing, but it starts off by saying, do you have planets there? If so, are they in the right place to have life? If so, is there water there? We do the water first, in part because it's easy to see. It's got big spectral features. If you got water, you start to look for the oxygen. You start to rule out the false positives, and it ends here. Do you have methane? If you have methane and oxygen, that's critical. And it's not, to me, a thermodynamic disequilibrium. It's more, in my mind, a kinetic one. The analogy I use here is if I see pizza in a room full of those same college students, I know there's a pizza delivery restaurant nearby. And the reason is college students eat pizza, okay? So if that pizza's there and it's in significant quantities and there's a significant quantity of college students around, you know there has to be a rapid pizza delivery mechanism, okay? And at least when I was in college, I wasn't making that much pizza, at least not in large quantity. Actually, I wasn't making pizza. I make pizza now, but I didn't make pizza when I was in college. I didn't have that skill set. So by the presence of the quantity of pizza and students in the same room, you can ascertain a pizza delivery mechanism, which is a pizza restaurant. It's the same thing. Oxygen and methane destroy each other. You can only keep them in the atmosphere together if you're replenishing them at very rapid rates. And those rates are orders of magnitude larger than any non-biological process. And all the non-biological accumulation ways to accumulate oxygen we know of actually work on the other end. They work on slowing down the destruction of oxygen so that it's slow enough that these slow abiotic production rates can maintain it in the atmosphere. So to me, the biosignature is about the fluxes. And to get the fluxes, you can't just make the measurement of the one gas you're worried about. You have to measure the entire atmospheric chemistry as well as you can. So we're thinking about these false positives. And we're thinking about what you would need to do to actually make the measurements to rule out the false positives. And one of the things that's quite powerful is the transit spectroscopy technique is absolutely tremendous, and it's going to tell us a lot about the chemistry of worlds in the near term. But eventually you want to do something like this. This is a wonderful movie that's a stitched together an interpolated set of images of uninhabitable worlds made from a ground-based telescope by Wang and Marois. And we want to do a movie like this for potentially habitable planets around other stars. Because if we if we do this, we're getting planet that's coming from the star, bouncing off the planet and coming back to our telescopes instead of seeing the planet pass in front of the star. And what that means is we get to see deeper into the planetary atmosphere, because instead of glancing through the top of the atmosphere, it's penetrating deep into the atmosphere before those photons return to our cameras. And what that'll let us do is it'll let us not just see deeper into the atmosphere, but also hopefully to the surface. If you have a big enough telescope and you look a long enough time with it, you can start to see the spectral features from the surface associated with oceans and continents. And if you stare at that planet, as the planet spins underneath the telescope, the planet's going to be giving you different sides of it as it rotates. And this was an experiment by Nick Cowan, who's now up in Canada, was at the University of Washington at the time, where he took a planetary spacecraft, pointed it back at Earth from further out in the solar system, basically convolved the data back down to a single pixel, and then looked at the pixel change over time. And he was able to tease out from the one pixel of light and its colors where continents and oceans were on Earth, because the Earth was spinning underneath that spacecraft and presenting different faces of the Earth over time. So he did the technique he used was a principal component analysis, and now we're looking at what we could do in terms of a principal component analysis for exoplanets. It was sort of a proof of concept. Now, to do this really well, it's going to take more than just a brilliant scientist or even a couple brilliant scientists. This is really going to take a whole community of people. I talked about what we've learned from the MAVEN mission, which is a planetary mission. It's really a heliophysics process. That's the loss of this atmosphere through the top. We have to think about these things in a system science context, which is something that the Earth scientists are teaching us, because you have to think about that biosignature in the context of volcanic production, in the context of oceans and continents. And this is where I have a lot of hope for our ability to do that. So this is a paper that just came out this year, where colleagues at NASA, up at the Goddard Institute for Space Studies in New York City, took an Earth climate change model, the same kinds of global climate models we use to predict anthropogenic climate change here on Earth. And they changed the inputs to that model to make it to simulate Venus. And they did it. This isn't modern Venus. This is an ancient Venus. They've also done modern Mars. But that tells me two things. One, the models are good. They're not overly tuned to Earth. But two, we have models that work out of the box that come with all that system complexity we need to treat these biosignatures in their planetary context. Unless you think this is just about NASA, we're actually got a whole community of scientists on board from universities and from across the country, and we're expanding this throughout the world. This is the Nexus Exoplanet Systems Science Group. We're bringing all this science to bear on the single topic across all 4 divisions at NASA. Because to understand this, we really need to think about these things as systems with multiple components interacting. And I like to close here for 2 reasons. One is I'm a geologist, and there's a history in geology seminars to close with a picture of a sunset. And outside of Dr. Wakeford's talk, this is the best thing I can do for an exoplanet sunset. But I also like to close here because when this movie was made, the astrophysicist said, well, that's nice and poetic, but that's impossible. And what folks like Dr. Fischer have found out is you can have planets around double star systems like this. They exist. We've found them. And we found a lot of other surprises too, and we're going to continue to find them in the near future on the chemistry of these worlds. What I want to spend the next 20 years of my life working on is working on the telescopes that'll get us biological signals from these planets so we can be surprised and rewrite the textbooks on how life interacts with its host planet. Thank you.
Okay. But also the thing that Shawn did, which I hope you got, was I think he made a very compelling case for investigating planets in a range of stellar environments, including ones that are more like the Sun-Earth system that we know so well. So now I'm going to talk about what will be needed to actually achieve those goals. So we are advancing different observational techniques and developing new technologies to get there. This graphic shows the arc of the past and near future facilities used for exoplanet studies, and I think every single one of them has been mentioned in the previous talks. And just to recap, the story started with the discovery of exoplanets using ground-based observatories, and Debra was part of that in the early days, still is. Those ground-based observatories are still critically important even today, producing more and more discoveries. So then Hubble made the first discovery of an exoplanet atmosphere. Then Spitzer and Kepler, well, Spitzer started characterizing the atmospheres further, and then Kepler just led to like an enormous explosion in a huge numbers of transiting exoplanets discovered, and really showed the huge range and diversity of their masses and their orbits. Okay, so then, as Hannah said, moving into the near future, the Transiting Exoplanet Survey Satellite will discover more small transiting planets, but this time they'll be orbiting stars that are closer to us, making them much easier for follow-up observations, which will be done with the James Webb Space Telescope, which should really knock it out of the park for giant spectroscopy, actually doing spectroscopy to characterize the atmospheres of giant planets and for some small planets around small stars. Okay, however, so we've done all that. That's what we've done. That's what's coming in the next 5 years-ish. But there's still an exoplanet parameter space that we cannot yet reach for atmosphere studies, and I'm going to talk about that a bit next. Okay, so again, just recapping again what Hannah said and what Shawn said. Searching for life on exoplanets is really a search for atmospheric molecules, atmospheric biosignatures. So we need to probe the atmospheres. Today, we're doing that for giant planets with transmission spectroscopy, as in this cartoon, light from the star. Some of it is blocked by the solid surface, and some of it passes through the atmosphere. If you obtain light curves at different wavelengths, as Hannah also said, you can recover the spectrum of the planet. So the point here is that different wavelengths of light penetrate more or less deeply into the atmosphere. So for example, here, the blue light ends up getting absorbed higher in the atmosphere, and the planet looks bigger, so you get a deeper transit depth. Then the red light penetrates more deeply into the atmosphere, the planet looks smaller, and you get a shallower transit depth. And if you do this for a bunch of wavelengths, you probe the depth into the atmosphere. So, and doing this, you can recover the wavelength-dependent atmospheric absorption spectrum of the planet. However, as you can see, this change in flux here, the relative change in flux, is proportional to the area of the planet over the area of the star. And so you can imagine this is much easier when the size of the atmosphere is large relative to the size of the star, like for a gas giant planet. The other issue with this technique is, as Hannah also mentioned, that clouds and hazes, as interesting as they are for their own sake, can obscure the lower atmosphere of the planet. And there's actually an interesting illustration. Actually, no, sorry. First I'm going to do this. Okay, so if we want to push this technique, so why can't we use this technique to do true Earths around sun-like stars. And here's why. There are 4 ways to reduce the size of that transit signal you're actually trying to measure, that dip in light. You could have a larger star / smaller planet. You could have a less puffy atmosphere, one that was colder or heavier. Clouds and hazes, as I mentioned, and then also refraction, just as the light just skims through the surface of the atmosphere and bounces off, like bouncing off a pond. All of these ways to reduce the transit signal, they all apply for Earth-like atmospheres around, for Earth-like atmospheres on Earth-sized planets around sun-like stars. So in sum, the spectroscopy of Earth-like planets, the ones that are more like us, we really need a different technique. In particular, what we need to do is we need to collect photons from the planet alone this time, not a huge bucket of star photons with a tiny little signal of planet buried in it. We want to just suppress the stars and actually just observe the planet itself. So actually saw kind of an interesting illustration of this that we just got from the New Horizons mission to Pluto. That shows kind of what I'm talking about here, I realized. So this is a view of Pluto's limb. New Horizons had passed Pluto and was looking back towards the sun, much like a transit observation. And it was actually super interesting because they saw for the first time, unexpectedly, this very extended haze layers, many of them reaching up in the atmosphere, which was actually very interesting, but for our purposes, haze is bad. And an observation like this is extremely sensitive to haze and cloud. Of course, Pluto's atmosphere is extremely tenuous, so you can still see the surface of the planet, but still, this particular observation was is sort of in a way dominated by the haze layers. On the other hand, this is New Horizons when it was on its way to Pluto, approaching Pluto. This is the sun side view. And this is analogous to a direct observation where the sunlight hit the planet and gone all the way down through the whole atmosphere, bounced off the surface and come all the way back out again. And this is more like what we would do, this is more like what we would want to do for exoplanets, what we'll need to do. Because as I think Shawn mentioned also as well, we actually do want to be able to probe all the way through the atmosphere, all the way down to the lower atmosphere, and actually try to get a hint of what the surface features might be. Okay, so why aren't we doing this? Why aren't we taking pictures of exoplanets around sun-like stars right now? Okay, because it's really bloody hard, and let's just try to wrap our heads around exactly how hard it is to take a direct picture of an Earth around a sun. Okay, so this is, Earth is 10 billion times fainter than the sun. So I tried to come up with an actually mathematically accurate analogy for that. And actually the traditional firefly next to the searchlight, not faint enough. Firefly is too bright. So the Luxor skybeam is the brightest artificial light in the world coming out of this hotel in Las Vegas. If that's the sun, then the Earth is 4 lumens, 4 standard dinner table candles. But astronomers actually observe things fainter than that. We've done that all the time. We do that all the time. We've done that before. That's not the really hard problem. The really hard problem is that those 4 candles are right next to the bright source. So for the Earth-Sun system at a distance of 10 parsecs, which is not very far away, the maximum angular separation, apparent separation between these sources is 0.1 arcseconds. And that is about the width of a human hair at the distance of 2 football fields. Okay, so now imagine the difficulty of trying to get the light just from those 4 dinner table candles when they were sitting right on top of the Luxor sky pyramid thing. Okay, so how can we do this? We need to dramatically suppress the light from the star so that we can see the pale blue dots next to them. And there are at least 2 different ways, technological ways, that people have thought of doing this. Okay, so, and here is the first one I'm going to go over. So this is a movie that shows how a high contrast instrument called a coronagraph works. Now these aren't new. We've had coronagraphs for many decades. They were originally invented to study the sun's corona. And they are used now already to take direct pictures, direct observations of exoplanets. Those 4 super Jupiters that Shawn showed orbiting their star, those were done with a coronagraphic instrument on a ground-based telescope. But we need to push this technology a good deal before we can actually do Earths around suns. So let's see, let's watch this movie, that very nice one that JPL made, with a bit of a intro that says everything I already just said. Okay, should have cut this part. Okay, now we go. Okay, so there's a telescope. There's light from the star coming in. And the wavefront is coming in, and it hits a number of optics. And if you don't do anything, that's what you see, an airy diffraction pattern. If you put in an occulting spot, basically a little shaped mask, you suppress, okay, and then a Leo stop, okay, it's better. And now you see, okay, nothing. All right, let's change the contrast. Oh, wait, first of all, imagine that there was a planet actually in there, and it's coming in at a slightly different angle. Okay, now we change the contrast, and we still see nothing. It's still just a bunch of residual speckles of starlight. So what really needs to happen is, if you notice here that the shape of the wavefront coming in from the telescope is distorted. It's not flat like it's supposed to be. So this is the new innovation here would be adding what's called a deformable mirror that actually changes its shape to correct for the distortions that were introduced by the primary telescope, and then flatten the wavefront so that you can actually get good contrast and start to pick out the planets from the residual starlight. Okay, so that's one notion. Okay, here's another one. This one is intuitive and seems a lot easier. It's called a starshade. Unlike the coronagraphs, these have never been used before, and this movie will actually kind of probably show you why. But in a simple sense, it's blocking the light from the star again, but this time, the thing you're using to block the light outside the telescope. Way, way outside the telescope. All right, so let's watch. Okay, so the starshade unfurls and deploys. For this particular configuration, this is a fairly small telescope, maybe 1 meter, and this thing is about 40-ish meters in diameter. So starshades are in general, several tens of meters in diameter. Then it unfurls, it flies away from the telescope in order to make it so that you could see the habitable zones of planets in the habitable zones of nearby stars. The planet-starshade separation has to be something on the order of tens of thousands of kilometers. Significant fractions of the Earth-Moon separation. Okay, but still though, it looks very intuitive because it's actually much more like what we do every day when we are trying to make a high contrast observation, like an airplane flying by the sun. Block the sun with your hand. In the starshade, your hand is outside of your camera, the eye, and it's actually, much more like what we do. And that movie makes it look pretty easy. However, the shape of that starshade is critical. It has to be exactly precise and correct. Furthermore, if you want to retarget this thing to observe another star, you have to move it over an arc that's tens of thousands of kilometers long. So as you can imagine, the retargeting times are extremely long, and they take a lot of fuel. On top of which, they can't really be tested properly on the ground. I mean, the thing is tens of meters in diameter, and your full-scale lab would have to be out at the moon, I guess. So anyway, so they have their challenges. They have their development challenges, but in principle, they actually are very nice. In principle, they can achieve higher contrast than coronagraphs can at smaller separations from the star. We just, at the moment, it's still kind of like in principle. Okay, so where are we going to develop these two technologies to the point where we actually need them to be? So the next step is to go into space with the first really advanced coronagraph, and that will be done on the WFIRST mission. So WFIRST is NASA's next strategic mission after the James Webb Space Telescope. It's a 2.4 meter diameter telescope with wide field imaging. It's primarily actually focused on dark energy studies. But in addition, it will also carry a first really advanced coronagraph that actually incorporates those deformable mirrors that I showed. But it is a technology demonstration instrument, so it is not at the performance level anywhere close to the performance level that we'd need to do true Earths around suns, but it would greatly advance direct imaging from space. And this is a preliminary multicolor imaging simulation of what WFIRST would see for, let's see, a sun-like star at 3 parsecs, pretty close, with a warm Jupiter at 2 AU from the star. And actually all of this haze of light here is light reflected off interplanetary dust in the system, which is coming from asteroids, actually. It's expected to be there anyway. And just as a shout out here, the simulation was made by Neil Zimmerman at Goddard and Maxime Rizzo, my postdoc, who's right over there. So, okay, but like I said, so WFIRST chronograph should greatly advance the chronograph technology. I should say there is some talk about possibly flying a technology demonstration starshade with WFIRST as well, as sort of like as a follow-on towards the end of the mission. But again, neither of those devices paired with the WFIRST telescope are going to get us there, and to be blunt, the telescope is just too small. So what do we want to really do? So, so NASA started a study, a 3-year study of a concept for another strategic mission that would come even after WFIRST. So it's called Large UV Optical Infrared Surveyor, or LUVOIR. Not the catchiest title, but it's just a working title. And this is a concept for a space telescope in the tradition of Hubble. So a truly multi-wavelength facility with broad science capabilities. Its aperture will be in the 8 to 16 meter diameter range is what we're studying. Full disclosure, I'm the study scientist for this effort, and Debra is the co-lead of the science team, and Shawn is the deputy study scientist. So we also envision it would have like a whole suite of imagers and spectrographs, a very diverse toolkit like Hubble does. And also, too, it's planned to be serviceable and upgradable from the get-go. We want it to last for decades and have the ability to answer questions in 25 years from now that we haven't thought of right now when we're designing it, which was key for Hubble. But on top of that, its probably key science goal would be to really spectroscopically survey dozens of small habitable zone planets, actually probe their atmospheres and see if any of them show signs of atmospheric biosignatures. Habitable zone, in particular, habitable zone planets around a wide range of stars, including sun-like stars. Okay, and this is a simulation of what LUVOIR, preliminary simulation of what LUVOIR would see. This is the solar system from 13 parsecs with a chronograph and a 12-meter telescope, Jupiter, Venus, and there, the pale blue dot that we're actually hunting for. Okay, so just to wrap up, we'll fill out the rest of the NASA mission swoosh here. So moving forwards to WFIRST, and then hopefully to LUVOIR, or a different concept that's quite similar to LUVOIR that's called HABEX. So there is a possible path stretching out over the next decades that leads to the first survey for planets that could actually be hospitable for Earth life. And furthermore, if that kind of life is not extremely rare, then we'll have a fighting chance of finding some. And so I'd say we are really in the exciting position of possibly answering an ages-old question about our uniqueness in the universe, or perhaps, actually even more likely, finding out if we're actually asking the right questions. So, okay.
When you have questions, put your hand up in the air, and a microphone will come to you, and I will point to you and call your name. Stand up, tell us your name, and ask a question. No speeches, just questions. Speeches come in the social hour. Okay, so there's a question in the back there. You have to hold it close to your mouth.
Hi, I'm Joseph Powers. My question was, I see that it's just ground-based observatories there, so where do next-generation ground observatories like the 30-meter telescope and the Giant Magellan Telescope fit into this scheme?
Okay, yeah, right. So that's an exciting question. As these giant telescopes, 30-meter class telescopes, are built on the ground, they'll need to correct for the Earth's atmosphere, atmospheric distortions, and so fancy AO systems are being developed, and I expect that there's going to be a lot of progress with those telescopes. But I see those telescopes as providing a bridge to the space-based telescopes. It's just impossible, I think, to beat the resolution that you can get from space.
And one of the first questions we ask ourselves when we design one of the next generation missions is, what do we think the ground's going to be doing? Let's not duplicate that effort, let's complement it.
Yeah, exactly.
Absolutely.
If we can do it from the ground, we don't do it from space. But what I guess I would add, though, what I think the ground-based extremely large telescopes, or ELTs, that are coming, they should be able to do direct observations of Earth-like planets in the habitable zones of M-dwarf stars. So in that sense, it's very complementary to a space mission like LUVOIR or HABEX, LUVOIR and HABEX would focus on the sun-like stars, and the ground-based facilities will focus on the low-mass red dwarf stars.
Of course, there are problems with the Earth's atmosphere and what it's made up of. We have a huge amount of water vapor in our Earth's atmosphere. That absorbs the wavelengths where we're looking for water absorption in these alien planet atmospheres. The same thing for sodium, the same thing for potassium, ozone, CO2.
Yeah.
Huge amounts of things in the Earth's atmosphere that need to be corrected and monitored over the course of the night, how those column densities are changing. So when we're trying to understand the atmospheres and the environments of these worlds, we really have to consider those when looking from the ground as well.
Okay, next question right here.
I am Chris Sharma. I am a guest here. Oh, can you hear me better now? I know you are very confident of finding a planet and some life on it. Is there any way to calculate the age of that exoplanet? Because Earth took about 4 billion years to evolve to what we are today.
So the answer for the planet is no, and then I'll turn to any of my colleagues because our best guess is going to be from measuring the age of the star.
Yeah.
And they know more about that than I do.
Yeah, the planets form with the stars, no question. And we're getting pretty good at dating stars now, figuring out at least if stars are very young, middle-aged, or very old. We can at least classify them that way. And most of the stars in the solar neighborhood have ages that are not so different from the sun.
Dr. Roberge, you mentioned moving star shields around potentially, and I wondered, you said that it's difficult to do because first of all, it takes a lot of fuel, and also it's moving them large distances. How do you even begin to do that?
Well, you put little engines on the starshade, little jets, rocket. Actually, I suppose ion electric propulsion, I suppose, would probably be the first thing to do. But any way you slice it, it's sort of a fuel-limited problem. But as you can, I don't know. I mean, I think moving the starshade all over the sky, is not insurmountable. That is an engineering problem that can be solved. But the real challenges, I think, are figuring out if it works as a system, making sure that it deploys to the precision that it needs to deploy. That shape of that starshade is no accident. It must be exactly right. And it needs to be, you need to basically deploy to, I don't know, maybe tens to hundreds of micron precision over a thing that's 50 or 60 meters in diameter. That is no joke. So there are some other issues about how they will work as systems that haven't been investigated yet as well, I would say, because it's really hard to test and study this thing on the ground. Not that Northrop Grumman isn't trying, but shout out there for Alberta.
And just to give you an example of how hard that is, on the James Webb Space Telescope, you saw the 5 sunshield layers. We have to align those because you don't want the telescope seeing the layer at the bottom, which is facing the sun, is therefore warm. And that kind of alignment is around, a few centimeter kind of tolerance for each of the layers. So you're talking about, many orders of magnitude improvement in being able to predict where everything will be when you deploy it.
Yeah.
Hello, my name is Alon Lanier. I do not work in the field of astronomy, but I did read a little bit about the coronagraph, and I do know that James Webb Space Telescope, as you mentioned, will have a coronagraph. Like, what kind of capabilities will that coronagraph have regarding exoplanets? And second, I've read about other things, like that the Subaru telescope just had a coronagraph, and this university in Caltech, they developed what is called high-resolution spectronomy. I don't know anything about these technologies, but if you could explain a little bit about those and how they might affect the search for exoplanets. That'd be great.
JWST chronographs.
So the James Webb Space Telescope coronagraphs were designed some time ago, and we measure the performance of coronagraphs in terms of their contrast. So as you heard from Aki, to see Earth-like planets around sun-like stars, you need to be getting to contrast levels of around 10 billion. The James Webb Space Telescope contrast is around 10 to the 6, possibly 10 to the 7, and we're working in the infrared, so we cannot see that close in. And it's really designed to do observations of gas giant planets at fairly significant distances from the stars, like Jupiter and Saturn in our own solar system.
Yeah.
So it will do a good job for the problem it was designed for. It wasn't designed to do this really tough problem of imaging Earth.
But one thing I will say about the coronagraph on James Webb is these Jupiter-Saturn-sized worlds at those kinds of distances are very important for our understanding of our solar system, our terrestrial planets, and our habitability. So understanding how those objects are shaped in other solar systems is really key for a full and complete picture of our own solar system. And that's why studying and understanding the atmospheres, the constituents of these giant planets, the dynamics in those atmospheres is really, really important, because it really does lead back to our own planet and our ability to live here.
And I'll just add one other thing real quickly. The other unique capability that the James Webb coronagraphs have are the mid-infrared imaging from 5 to just about 30 microns. That we've never been able to do before. So that's a really new capability. It opens up a lot of molecular lines that we haven't really studied before.
Yeah, but as you said, those coronagraphs don't have deformable mirrors. So
The high-resolution spectrograph technique you mentioned, that's mostly being developed for the ground at first.
High-resolution cross
dispersion.
Dimitri stuff. So but we are looking at incorporating that into these future telescopes as well.
Yeah, that's a new technique that there's some things to still figure out, actually, on how to make that technique work at the really, really sensitive levels that you need for Earths around suns. Probably it can't do it by itself, but it might, combined with the coronagraph, it might actually give it a little extra oomph. So, but there's a lot still to study about that particular technique, the one that you mentioned, the high-resolution technique. So.
I wonder if you could say something about a different concept of telescope, where it's me again. Instead of these telescopes where you can just reuse it and point it at different stars, if you had something more like a sacrificial telescope that you send out to the extreme edge of the solar system and use the mass of the solar system as your lens with gravitational lensing. If you had one target that you really wanted to look at very closely, like in terms of the feasible technologies, what could you see with a gravitational lensing telescope? Aside from the politics of needing to send an actual nuclear reactor and not rely on space probe grade plutonium, but put that aside.
I mean, I can say a little bit about that. I was talking to some of my colleagues at Caltech who were talking about that idea. Yeah, so you send a spacecraft out to, as you said, to the outer part of the solar system, then you look back towards the sun, and because the sun has quite more mass than anything else in our solar system, as a star behind the sun, as that light's coming towards us, normally you might think, the sun will just block it out. But in fact, some rays of light from that background object are bending. We would have missed them. But the mass of the sun warps the space-time continuum, bends the light back to us, and focuses it so that we could actually, for the right alignment, and it would be totally serendipitous. It's difficult to plan this. You would be able to see with incredible brightness a planet orbiting a star that's behind the sun. You have to have exact, sort of the distances between the sun and that background object are important, and the distance of you to your spacecraft in the outer solar system are all important. But it does have the potential, right, to hit the jackpot. Oh, you would be able to get spectra. You might even see a bright enough image of a planet to see clouds rotating on the surface of the planet. It's a Hail Mary.
Hail Mary, invest in study.
I'm Patrick Alsop, and I've got a couple questions. One of the things I've read repeatedly about affecting the suitability of Earth for life is its rotation rate and the presence of a large moon. Is either of those, and actually I suspect that you probably can detect something like the rotation rate of the planet based on that surface survey, but could you detect a large moon or other characteristics that might contribute to the suitability?
The answer is yes, although it's going to be easier to do at some wavelengths than others. Actually, if you look at the wavelengths at which carbon dioxide absorbs or water absorbs, which our atmosphere is full of, the Earth can become quite dark. And the moon, because it doesn't have an atmosphere, retains its brightness. In the Earth-Moon system, the moon, I think, contributes something like 10 to 20% of the total brightness of the Earth-Moon system. So the pixel would get very dim at those wavelengths that water and carbon dioxide have deep absorption features. And at that point, you would actually be able to be getting essentially a spectrum of the moon around that planet. If you're interested in this, there's work by Ty Robinson and colleagues that came out a few years ago. So if we, again, looking at the time resolve thing and looking at the right wavelengths, you could probably tease that out of the data.
Problem is, of course, if your moon is airless, that's great.
Yes.
If you've got anything else, then
that's right.
We're very much based in that simulation situation on what we know here. Anything else will result in different patterns that you would see, amplification of signals at different wavelengths based on separate atmospheres. And that's certainly an area that people are exploring because it's really an interesting concept of whether or not we can discover and analyze moons around planets, including moons around giant planets, as we have around our giant planets in our solar system. So it's a tough question, but it's not without possibility.
And actually, I guess I would make one more comment too. I think the problem is maybe perhaps a bit backwards. I think we'll need to find the habitable planets, characterize them, see if they have liquid water, and then see if it matters whether they have a moon or not.
Yeah.
Because the importance or influence of the moon on our planet it's a data point of one, and it's all a bunch of theory anyway.
But now it's a testable hypothesis. We want to make it a testable hypothesis.
Yeah, it's a testable hypothesis, but I think that it's the other way around. We'll find the habitable conditions first, and then figure out, well, what are the factors that affect whether this planet is habitable or not that one or something. And moons may be a factor, but I doubt it's the most important one. I really do.
Mm-hmm. Yeah, I think exoplanets it's been astonishing. Theory has been wrong almost all the time. And it's really the empirical evidence that just has left us saying, what? How can that be?
Yeah, it's not even just scientific theory. It's our imaginations aren't as good as nature's imagination. Yeah. They're just not. Science fiction did not predict the presence of giant Jupiter-sized worlds so close to their star, 20 times closer than we are to the sun.
Yeah, even sci-fi didn't predict
Sci-fi couldn't work it out. Our imaginations aren't as good as Mother Nature, and we're going to be finding out exactly how far that goes over the next couple of decades.
Yeah, and I mean, my personal research is in planet formation. Off the top of my head, I can think of like 4 different ways to prove from theory that planets cannot form. Right.
Okay, Carl Merrill, I'm a member of the Society. Could you clarify what you think would be a real signal of life? Because on one hand, I thought oxygen was a signal. On the other hand, you said, well, oxygen could be formed without any life. On the other hand, we know on this planet there was life before there was oxygen, before the cyanobacter started making all this extra oxygen. So I'm not really sure. And then the other question along the same lines is, could we get a signal from complex organic molecules when we're doing the spectrum?
So apologies for muddying that one up a bit. Both in the literature and in this talk, actually. Let me give the specific answer and then the general one. For modern day Earth, the best biosignature is still oxygen and methane together in the presence of liquid water. I think myself as a scientist, that's the point at which I'm willing to call the president, call the press conference, write the paper, and go forward. You know, if the rest of the scientific community disagrees at that point, that's on them, you know, or that's between me and them. But like, that's when I'm willing to put my name on the paper that says, yeah, we found evidence of life elsewhere. More generally, and we're working actually, there's going to be papers coming out next week that suggest that we actually turn to a more quantitative approach using Bayes' theorem. And to do that, you need the prior, which is the whole point of doing the mission in the first place. But you also have essentially the ratio of the probability the data arise that you see from a living planet versus one that is habitable but not alive. And to get that ratio, we actually have to build really good models of planets that are alive. Like, we have those actually. Our GCMs are great models of planets that are alive. We also need to develop really good models of planets that could be alive but are not. And that's actually, I think we're actually behind on that one. That one's actually more difficult to do because life is so intertwined with the one planet that we know that has the conditions for life. That'll allow us a more quantitative assessment. I think there's other things I could say in general terms, like, you know, you want to look for a motivation for life to be doing something, some sort of metabolism to get energy from, and then detect the byproduct of that metabolism. Those are the kinds of things that would be very, very strong indicators of life. And that thing that it's producing can't be produced abiotically at a fast enough rate to accumulate to a detectable amount. So, but we need to be more quantitative, and I think over the next 5 to 10 years we'll get there.
Yeah, I think in the 10, 15 years ago, it was kind of put rather as a simple observational problem. Detect oxygen, you're good. Then it got to, okay, well, all right, you need to detect oxygen and methane. But now I think the real answer is, like, you need to try to measure all the abundances of all the primary constituents of the atmosphere and figure out what all the sources and sink rates are. And only when you see something that is anomalous, that's not physics or chemistry, biology.
Although it's important to have the motivating thing, because I don't believe in a life between the gaps method of finding life, right? Okay. There needs to be that, you know.
This isn't totally settled yet, by the way.
I mean, to me, if you didn't know the biosignature was there, and you said life is on this planet, what would it be doing? You should predict the thing that life would be making the thing you think is the biosignature, right? If you have a planet.
Identify a food and a waste product.
Yes.
Okay, fine.
Predict the waste product that you're identifying.
Okay. We haven't completely figured all of this out yet. Okay, that makes sense though.
Those your questions answered,
Carl? The complex organics. Oh. So there's a paper we have in review right now that suggests that the hazes that Hannah wants to look at, that some of them that we think existed or were present on ancient Earth, in certain atmospheric contexts can only occur if there's biology making a lot of methane. Now, the problem with that is there's plenty of non-biological ways to make methane. And the biosignature might be, and this is a might, because this one we haven't thought about the false positives yet, is when biology makes methane, it also releases a bunch of other organic compounds. And those organic compounds are more easily split apart by photolysis in the atmosphere and by kinetic reactions in general. That is going to facilitate haze formation much more readily than methane that's coming from non-biological sources. And so if you've got the haze and you can get that atmospheric context to know how fast the haze could be produced with just abiological methane production, you might be able to say something. But the individual complex organic molecules are probably not going to accumulate to a high enough concentration to detect. We'd only see them in mass if they're making these large spherical droplets that form hazes that even Hannah's detecting now in the gas giants.
Yeah, and you know, if you do detect those kinds of things, it tells you a little bit about their dynamics, but it's not a you can't get much more about that planetary atmosphere beyond something has to be lifting it up and sustaining it at that altitude. So it gives you some things, but it really hinders a lot of other investigations.
Have a Europa or Martian scientist come and ask that question again. You'll get a good answer.
Titan.
Or Titan.
Titan.
Isaac Schomer, I'm a member, and my question is about, in the third lecture, there was something about hydrogen in the atmosphere that was being blown away or lost, basically. And I'm wondering if some of that hydrogen could be regenerated by protons coming in from the stellar wind.
Yes, and it could also be regenerated from volcanoes at the surface of the planet. And we're a bit paranoid about these false positives because we don't want to do the thing I started off talking about, spending our whole lives on these careers, not to mention a considerable amount of taxpayer dollars, only to have the wrong search strategy. So we're being paranoid, and we're trying to concoct planets that'll make the gases we think are biosignatures. So you're right. You would need to not be accumulating hydrogen from that stellar wind. You also would need to not have volcanoes putting hydrogen into the atmosphere like volcanoes on Earth tend to do. So you kind of need the just right planet. But I don't want to rely on the just right planet not existing before we go forward with a mission like this.
So we have one there, and then
Yeah, Bob Abramson, member. From the standpoint of looking for intelligent life that might inhabit some of these exoplanets, any thoughts on the collaborative use of radio frequency astronomy in ways that might either guide searches or, if we do hear signals that appear to be of potential interest, to try to correlate those against specific observations with Webb or other resources?
So I think there's an interesting story here with, there's a history of NASA of not funding much SETI work. But there is one grant that NASA has out to fund a little bit of radio telescope observations. And that's for some of these Kepler worlds, which as Dr. Fischer mentioned, they're too far away for us to do the direct imaging spectroscopy that we'd like to do to look for chemical biosignatures in the atmosphere. So the only next step you can take to test the hypothesis of whether or not they have life is to do a search for radio signals. And so the one sort of small part of SETI that's NASA funded, that, to my understanding, is to do just that with Green Bank. And I think if we found any of these worlds, then yeah, of course we'd be looking at what signals might be coming from them. I actually think that that's a better time to do it when you have a more directed hypothesis to be testing. That's just me personally.
Hi, Marshall Eubanks, a guest. You said something interesting that caught my mind, and I didn't quite understand it. You said instead of looking for chemical disequilibrium, we should look for kinetic disequilibrium.
Yeah.
And I presume you're not talking about bullets here, so what are you talking about?
What I mean by that is kinetic chemistry as opposed to thermodynamic chemistry. And the difference is thermodynamic chemistry is essentially it's the difference between the energy that you could get from having those two molecules in the atmosphere together. Sort of, you could make a measurement of how much energy you get from reacting them together. Two molecules that shouldn't be in the atmosphere at that temperature and pressure. That's the thermodynamic argument. Personally, I don't like that argument, because if there's something that biology could get energy from in the atmosphere, I don't think that's a good biosignature, because the biology should be getting that energy from that source. We actually just saw a workout on Enceladus along these lines, where they detected hydrogen, which is great because it's an energy source, but it's not great because it's an energy source, and why isn't it being used if there's life there? The kinetic one is more just about production and destruction rates. If it's being destroyed really, 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 really fast, then it has to be replenished really, 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 really fast. And if the replenishment rate is orders of magnitude greater than any of the non-biological production rates, but consistent with what you would predict from biology, then you're onto something, I think.
Practicing swallowing the mic. Frank Robert, a member. I don't envy you the task of distinguishing the tholins on Titan equivalent on an exoplanet from those of life, but probably an off-the-wall question. Would the use of a background star for occultation thinking of the discovery of the rings of Uranus and so forth, be feasible from at these distances, or is this complete pie in the sky that you could image that way?
I'm going to punt.
Oh, a background star, an occultation, a background star occulting a planet, not the host star. A background star, yeah. Well, the problem with that is to know when that's going to happen, you have to have the planet's orbit. So you have to find it first. You have to find it first, and oh Lord, I actually did some work in eclipse and occultation work when I was an undergrad with Jim Elliott, actually, and that is, yeah, that is a major pain in the butt. And you would have to know the planet's orbit to exquisite precision and the proper motion of the star itself, the host star itself. You'd have to know every piece of orbital information to like crazy precision for that to even have a hope, I think, for that.
Makes star shades sound easy.
Yeah, yeah, yeah. That's yeah. I think there are easier ways of doing this than that.
Number 2 for you. And then we have another one.
Yes, my name is Alon Lanier. I am not a member. Just each time they've discovered planets that caused excitement in the news, like the TRAPPIST system, and before that the Proxima Centauri system, we got cold water because we're told that the M-dwarf stars are too active and cause solar winds that cook the atmosphere. You alluded to that today. Do we have any candidates out there left that their stars are not too active, that James Webb that will fall within the range of James Webb? That's like if you'd had to guess which is the best candidate, what would your guess be?
So the TRAPPIST system is still by far the greatest candidate for these kinds of observations, certainly with the James Webb Space Telescope. Kepler has discovered a number of planets which are around larger stars. So the TRAPPIST-1 star is much smaller than our sun. So around larger stars that are in the habitable zone. Younger stars rotate quicker and are more active, so that's an issue as well. So we need to understand the ages of these systems. Unfortunately, most of the Kepler planets are very far away and very dim. So when we're pointing our photon bucket at these stars, they're not collecting enough, and certainly not enough that's shining through those atmospheres. So the atmosphere is 1% on 1%. We're looking at a point source on a point source, and we're trying to work out what the environment's like. So for the Kepler targets, it's very difficult to do. You'd need something like LUVOIR, which is why that kind of instrument is so important. That size of telescope, that kind of instrumentation that will be developed for that kind of telescope is going to be amazing for these kinds of studies. But the M stars, we're trying to really understand those and trying to understand the environments they do cause on these planets, because we know loads about our sun. It's right there. We've even thrown a couple of things at it. And we're trying to understand other stars and how that impacts the possibility of life. So these types of stars and looking at different types of stars and types of planets is really key for that.
I think we'll have one more question before we move on to, okay, two.
Thank you. I'm Joe Shulman. I'm a member. You know, Carl Merrill asked a key question, in my opinion. Imagine that you had some imaging device way out in space, and you wanted to look at the Earth, where we know there's life, and ask the question, how do you know there's life on Earth? It seems to me that about the only marker you could think of, at least that I can think of, that for sure would tell you there's life on Earth would be the spectrum of chlorophyll. And, you know, because all these other things, you know, can arise in all sorts of ways that have nothing to do with the proof of life. That's my question.
So there have been studies of Earth as an exoplanet, trying to look at, so we can look at the Earth's light that's filtered through the atmosphere by looking at the moon as we eclipse the moon. We can study the Earth's atmosphere in the same way we're studying these exoplanet atmospheres and looking for Earth as an exoplanet. And a number of other things have been done to that effect, trying to understand what our spectrum looks like on a global scale and how much we can resolve from that. Shawn can probably talk more to the chlorophyll, but...
Can I just say my piece about the red edge?
Yeah, say
Go
it.
ahead, give the bad news, then I'll give the good news.
I'm going to give the bad news on that one. Yeah, it would be great to detect the red edge, the photosynthetic red edge, spectral red edge. That would be fantastic. The problem is it's extremely weak feature. It is observationally probably pretty much, if we were looking for that feature from interstellar distances, we just could never see it. I mean, you can't even see it in the Earth shine. When the Earth's light, sunlight hits the Earth, bounces up to the moon, to the shadowed part of the moon, bounces back down into the telescope. This is like a poor man's exoplanet spectrum. Even in those Earth-shine spectra, you can't see the red edge unless it's a clear day and Brazil is facing the moon.
Although
you can really only see it. You would really only see it in like some sort of jungle planet that was completely covered with vegetation, and even then, there's no guarantee that it would be exactly at that pigment, that light-absorbing pigment would be at exactly the same wavelength as Earth's chlorophyll is at. And so how would you ever know that's what you were looking at? So that's the bad news.
But Brazil, the good news is, sometimes Brazil does face the moon. So that map I put up there where I said the planet spun underneath the planetary spacecraft, the author of that paper, it did not put this in the paper, but he'll tell you over a beer that the three dominant signals in the data that he pulled out of the principal component analysis correlated to continents, oceans, and clouds. And that's what's in the paper. What's not in the paper is the fourth major signal, which he couldn't quite detect to a significant degree, was actually a chlorophyll. It was a pigment signature.
Green.
Now,
it was green.
Now, I think because of the
It wasn't chlorophyll, it was green.
but it correlated to where chlorophyll exists on Earth, right?
Yeah.
Now,
also the peak output of the sun.
Yes.
So I think with all of this, it's not the thing you build the telescope around.
No.
But you want to the telescope should be able to try to see that, because if you see the oxygen, you see the methane, you see the water, you see continents and oceans. If you stare at it more and you start to see the hints of a red edge feature, I do think that's better than not having the red edge feature at all. Personally,
there are plenty of mineralogical species that have edges in their spectra too.
Yes, there are. Yeah, it's not the first thing you look for, but I think of it as a secondary biosignature.
Tertiary.
Clouds are really... Clouds are important.
I really think it's a red herring. I genuinely think it's actually a red herring. Red edge is a red herring.
A red edge herring.
I want to just remind you of the history of exoplanets. When in the early 1990s, people said, this is too hard, we're not going to find planets. They probably don't exist around other stars. And what we found just completely revolutionized the way that we look. So right now, we're going into this with a bias of what we think life might be like. We could be completely shocked. It could be much harder than we expect, and it could be much easier than we expect.
The problem
but
is
we
that,
have to look.
Yeah, the problem with this particular one is that we are biased on both sides.
Right.
We have it on Earth. Your bias is that we have it on Earth. It's a sign of life on Earth. My bias is, but we can't see it even when we're looking at the Earth. So we are both still Earth-focused on this.
I mean, this comes back to what Shawn was saying about we need to put simulations which can't make biological Earths. We need to understand how to do that, and we've got to be asking the questions without those biases which are just inherent in our brains from birth.
Yeah, that's actually
tough. It's a really tough thing to do.
That is a genuinely fascinating question. We have no idea what a habitable planet that's not inhabited actually looks like. Yeah.
Hi, I'm Jim McCormick, and I am a member. So last question, feel free to just say no. There's no doubt that there's a lot of pure science in what you're doing, but where you are, you have to be just really in tuned to budget to aspiration, right? There are social impacts on what you do and social significance, which is why you do what you do. Can you say something not obvious about the social impact on what you do, whether it's
inspiration.
Science, national, global?
This is an inspiring topic. It's the first question that man has ever had, looking up at the heavens. That's how we were defined, by looking up at the stars and trying to really understand and use them to live on this planet. this is an inspiring topic, and it generates people and in young children to go into science, to understand more, to go into engineering, build that telescope. The people that are building the telescopes, and Mark was telling me this earlier, they're so excited to be just part of this big, big mission, this big question that we all have inside of us. And that is so important, and I don't think that should be ever understated.
But what if you turned it around? And what if actually I have the answer in the back of the book, and I can tell you that among the 200 closest stars, that 75 of them have planets with life on them. And I know that that's true. Would we just not bother to look? So for some people, maybe the answer is yes. Maybe that's not what they're interested in. But I think for most of humanity, right, that it's just too compelling and too interesting. If it's really there, we have to find out.
And I guess I'll add too that I have a view that, like, you know, I don't intend this species goes out when the sun does. You know, someday we need to find new homes, ones that are nicer than Mars.
Mars ain't bad.
It's not bad.
No, it's
Wow.
not nice to hear. Somewhere
That
nowhere
is a
it's nice
big
and-
thought.
For me, I want to know how planets work, and I want to know how rocky planets work. And I think that the more sample size we get in understanding how, especially rocky planets that could harbor life work, I want to know how those things operate. We're finding out a lot already about how planetary systems work. Hannah's finding out a lot about how gas giants work, and is going to be finding out about whatever increasingly smaller planets do and how they work. But eventually, I want to know, like, how every time there's like one of these planets, like we have these like nerdy conversations at lunch, like, oh, that's going to test the carbonate silicate feedback cycle, which is like a major Earth sciences hypothesis. I want to test that on these exoplanets and every other hypothesis we have.
Yeah.
The secret to this is, 50 years from now, the press headlines will be written about whether or not we found life. But I think all the scientists will be remembering this as the comparative planetology mission that got us 500 worlds to compare our own solar system against.
Yeah.
So you've seen how engaged these folks get in this subject. At Goddard, we do hundreds of tours every year for various STEM programs, kids from 5 years old to 15. And the one thing they all ask about is this. So it's just the best hook for getting kids engaged in science and starting to think about the world they live in and the universe they live in.
So I'm going to add a note to that. I think we all think the great thing about science is what we know, but actually the really great thing about science is what we don't know and what we find out. And I'll quote Frank Zappa. How many of you know who Frank Zappa is? Okay, most people know Frank Zappa was a rock and roll musician and a little bit of a philosopher. And he quoted Edgar Varese, I think, who said, the contemporary composer refuses to die. And I'd say the contemporary scientist refuses to die. And what he was getting at was that you always have the contemporary scientist or artist who's after something that the rest of us don't really get because we're not out there on the edge. And this is what creates knowledge and creates beauty. And that in and of itself is more important, I think, than an engineering project that might be designed to achieve some practical goal. So I think, in the end, this is how we create our future. And there's really probably no more interesting question than, are we alone? And I think, I agree with Debra, there's probably a lot of life out there. And there's a lot to be learned by looking for it. So I'd like to thank our speakers. And before I let them escape, I have to give them a little present to thank them for their time and their expertise and for opening up new worlds for us. And that is a framed copy of the announcement of this Joseph Henry lecture. And I have one for each of them, which I'll give them in a minute. And I also have a copy of the Volume 1, since they're all members now, and signed by me, so it's a special one. So if you can hold it facing the
Oh.
audience.
Oh, I should apologize to that guy.
Before
for
you
going
go,
off
there
on
are a few
Reddit.
closing announcements.
Just go. God.
First and foremost,
Sarah, so many years.
thank you to all of our members, sponsors, and supporters.
Possible.
If you're not a member, please apply for membership, which is easy to do. Just go to our website and click on the membership button, and that'll bring up a screen. Fill out the form and hit the submit button, which will bring up the dread PayPal screen. So just ignore all these references to PayPal, because PayPal is just serving as our merchant bank to process your payment. Go down here to the continue, which brings up your usual payment screen, and you can make your payment using your favorite credit or debit card. This is our last lecture of the Spring 2017 lecture series. The fall will begin this year on October 6th. Please check our website, www.pswscience.org, for updates to the schedule, which hopefully will be completely posted by the end of the summer. The social hour ends at 10:30. Please exit, well, actually 10:45 tonight. Please exit by the side doors. I will now accept a motion by a member to adjourn the meeting. Second? All in favor? All opposed? The 86th Joseph Henry Lecture is adjourned.